When we look at the mitotic phase, okay, interphase has completed. We're now moving into M phase. The mitotic phase is also known as karyokinesis. Karyokinesis literally means DNA movement. It's the nuclear division. Not the dividing of the nucleus itself, but the nuclear contents, the chromosomes. So when you look at, in this textbook, you look at other textbook, other papers, you'll see karyokinesis, or you'll see it referred to as mitosis. Potato, potato, same thing. Mitosis, its whole point of being is to ensure the equal distribution of chromosomes to the daughter cells. Now, I've already used this term a couple of times. Daughter cells. Daughter cells. One cell divides, becomes two. The two are the daughter cells. The original is the parental cell. When you look at the microscopic photograph, you can see different cells in this, I believe it's a plant tissue. The cells are going through the different phases of mitosis. You see chromosomes condensing. You see them lining up. You see them being separated. You see the cells resolving. Mitosis is going to quickly be followed by what is referred to as cytokinesis. Basically means cell splitting. Once the chromosomes are divided, equally divided, nuclear membrane starts to reappear. Once the nuclear membrane starts to reappear, the cell will divide itself. Exactly how we'll talk about in a couple of lectures. So when you look at mitotic phase, you'll notice that there are 5 distinct phases followed by the cytokinesis. Prophase, prometa, meta, ana, telo. Cytokinesis. Prometa, meta, ana, telo. Prophase, prometaphase, metaphase, anaphase, telophase. Cytokinesis. Now what we're going to do over the next couple of lectures is we're going to go through each of these phases and we're going to discuss what are the characteristic actions, characteristic events occurring during these phases. When we start off here with prophase, the cell has immediately just left G2, it's entered into prophase. During prophase, the nuclear envelope disappears, breaks down. Machinery within the cell breaks down the lipid bilayer that makes up the dual membranes of the nuclear envelope, breaks it down and moves it away, or moves it out of the way, I should say. Large organelles, the ER, the Golgi, and whatnot, also get broken down, get shuttled, get moved out of the way, because, if you look above my head here, you'll see that this is going to take up a large volume of the cell. Centrosomes, which are large microtubule formations, these begin to form. They begin also moving to opposing ends of the cell. These are going to act as anchor points. Anchor points so that the chromosomes can be pulled apart. You need these centrosomes, these anchor points, because think about it. Last time you were standing on ice or you were floating in a pool, did you ever try to push somebody? Well, you push them, they move, you move. Okay? You can't pull apart stuff if everybody's able to move around freely. So, you need to have something, you need to hold on to something. A fixed point that won't float away or won't slide across the ice. That's what the centrosomes are. They're fixed anchor points so that the cell can pull the chromosomes apart. You also begin to see what's referred to as the spindle form. These are projections off the centrosomes that will attach to the chromosomes that will allow for the pulling apart of the